Let's take a look at gene expression regulation in eukaryotes. So we had looked at prokaryotes already, since the main difference between prokaryotes and eukaryotes is that prokaryotes do not have a nucleus. And because they don't have a nucleus, that means translation is not separated from transcription. So translation actually starts before transcription has finished. That means that in prokaryotes, the only way of regulating gene expression is by regulating transcription, so the binding of RNA polymerase to the promoter region and then being able to access the coding region and transcribe it. But eukaryotes have many more options for gene expression regulation. And this is for multiple reasons. Not only eukaryotes have to respond to the environment as prokaryotes do, but most eukaryotes are also multicellular organisms. So you have multiple types of cells in one organism, and all those cells derive from one single cell that was the original cell of that organism. So all multicellular organisms started as a single cell, and then that single cell divided many times by mitosis, which creates identical cells with identical DNA or virtually identical DNA. And then those cells with the same DNA had to differentiate and become different cells for different tissues. So even though all of them have the same DNA, they still have to perform different functions. So that means they have to express different sets of genes. And this is not just at one point in time, but this happens throughout the entire life of the organism. The cells will be adjusting and producing different proteins depending on what is needed for the organism at that time. So in eukaryote, cells need to respond not just to the environment, but they also need to respond to the developmental stage of the organism, as well as cells in different tissues need to perform different functions. So they need to produce different proteins, express different genes. So this process starts since the very beginning. Like we said, all multicellular organisms, either plants or animals, start from a single cell, and that single cell divided multiple times. And then slowly, those cells started differentiating towards the specific tissues that they would eventually become. So as the process of development happens, not only you are growing in size as having more cells, but also those cells are slowly becoming more and more specialized into the particular pathway that they're going to take. And so when that is happening, their patterns of gene expression are changing. So cells that are going to be part of the digestive system, for example, will turn off genes that are only expressed in the nervous system. So since they're not going to be a neuron in the future, they're turning those genes off. So how are cells able to respond to the developmental stage that the organism is in, to differentiate themselves, to have the particular gene expression pattern of the type of cell, of the type of tissue that they're going to belong to, and also to be able to respond to the needs of the organism in a particular point in time. Like if you're running out of energy, then you need to produce more of the enzyme glucagon that will take the reserves in your liver and turn that glycogen into glucose that you can use for energy. That needs to be coordinated when you need to express specific genes. All the possibilities for gene expression in eukaryotes happen because of the multiple steps that are required in eukaryotes to go from the information in the DNA and be able to use that information to make proteins. Starting with the chromatin, in eukaryotes, the DNA is not just by itself, but it's actually wrapped around proteins that are called histone proteins, and that complex of protein and DNA is called chromatin. The shape of that chromatin can be altered, and that can affect gene expression. As in prokaryotes, we also have transcription regulation where you need RNA polymerase to be able to access the promoter, but in eukaryotes this is a little bit more complex. In eukaryotes, since you have a nucleus and transcription is happening in the nucleus and the ribosomes are outside in the cytoplasm, you have time to do modifications to the transcript from the time it is made to the time it is exported out of the nucleus. And once the RNA is exported into the cytoplasm, you can also do regulation between transcription, and once the protein is made, you can also do additional modifications that might not happen in prokaryotes. So we have lots of places where gene expression can be regulated in eukaryotes, and we're going to take a look in more detail at what those are. So let's start with the beginning, before even transcription starts, so before even RNA polymerase can access the promoter region, we need to look at the structure of the chromatin. The DNA in eukaryotes is wrapped around proteins. Those are called histone proteins. That complex of protein DNA is called chromatin. If the proteins are very tightly packed around the DNA, they make very tight chromatin, and that makes the DNA unavailable for RNA polymerase to find the promoter. So if it's all tightly packed, RNA polymerase cannot access that DNA and cannot transcribe it. If, on the other hand, the histone proteins are loose and there are spaces between the histone proteins so that the DNA can be accessible to RNA polymerase, then transcription could be possible. And this regulation of gene expression by altering the structure of the chromatin is called epigenetics. It's called epigenetics because it is not changing the genes that you have, it's not changing the structure of the DNA, the sequence of the DNA is the same, it's just changing how tightly packed that DNA is or how loose it is. And the way you can go between back and forth into these stages, one way is using DNA methylation. This is here, adding methyl groups to the cytosines. And when you add those methyl groups, it can either make it unlikely for RNA polymerase to bind to that if that's a promoter region, or the other thing that happens when you have those methyl groups is that the DNA now packs more tightly around the histone proteins, so it becomes more compact, which means it's less likely to be accessible for RNA polymerase to transcribe it. So DNA methylation will decrease the chances of those genes being expressed. The opposite to that, to counter that, would be histone acetylation. Now you're adding an acetyl group, but now that acetyl group is being added to the histones, not to the DNA. So when the histones do not have acetylation, so they are unacetylated, they're very tightly packed around the DNA. If you add acetyl groups to the histones, as you see here, these dots, green dots are acetyl groups adding to the histones, that makes them lose the grip on the DNA, so they loosen up the DNA, and now the DNA is accessible for RNA polymerase to come and transcribe it. So when you have histone acetylation, that loosens up the DNA and makes them more likely to be transcribed. You also have enzymes that remove those acetyl groups, so that will be called histone deacetylation, and that will move in the opposite direction. So it will make DNA that was loose, as you remove those acetyl groups, it will become more compact. So histone acetylation makes the DNA looser and more likely to be transcribed. What's interesting about this epigenetic regulation is that it is very sensitive to the environment. You can have the same DNA, they have identical twins, for example, but they will have different patterns of DNA acetylation and methylation depending on the things in the environment that they're exposed to. And what is very interesting is that our epigenetic changes respond to very diverse type of stimuli. So for example, you respond to the diet, you respond to if you have different disease exposure or chemical exposure, like if you smoke or use drugs, or even your financial status, your emotional status, the relationships that you have, even the microbiomes that you have in your gut, so the type of bacteria that live in your digestive system, all of those things affect your epigenetic patterns. So all those things can affect the expression of your genes. So epigenetics is an incredible way for adjusting your gene expression to what is happening in your external environment. And this is a focus of research where very interesting things are being discovered here. So now that we saw how the structure of the chromatin can impact whether the DNA is accessible or not for transcription at all, now let's look at the regulation of transcription per se. So that is, once the DNA is available and proteins can bind to it, how do you determine whether that DNA will be transcribed or not? Compared to prokaryotes, where RNA polymerase can bind to the promoter directly with its signal subunit, here in eukaryotes, RNA polymerase cannot bind to the promoter directly on its own. It needs a combination of transcription factors in order to bind to that promoter. So you need to have the right set of transcription factors so that RNA polymerase can then bind to those transcription factors and start transcribing the gene. So one way of regulating gene expression here is by regulating the availability of those transcription factors. If you don't have the right set of transcription factors in that cell, those genes will not be expressed. And just like we had in prokaryotes that we have activator proteins, eukaryotes also have activator proteins. And so the right set of activator proteins need to be present, and that will make transcription even more likely. And you can have some proteins that might block transcription factors, and those will act kind of like repressors in that they would prevent RNA polymerase from binding to the promoter region. Let's say we have the right combination of transcription factors and enhancers and other proteins that we need in order to transcribe that RNA, and the RNA is being made. Since this is happening in the nucleus and the RNA still needs to be exported out into the cytoplasm for translation, we have that time and space in the nucleus to still do other modifications to that RNA transcript before it goes out. One of those modifications is splicing. So in splicing, the mRNA transcript has parts of the sequence that won't be expressed and won't be translated into protein, and those pieces are removed. The sections that are removed we call introns, as they won't get expressed, and the pieces that remain are called exons, as they're the ones that will get expressed. But what's interesting is that in multiple cells, or cells at different point in time, can remove different sets of introns and leaving different sets of exons. So for example, in this diagram here, we have these 4 sets of sections in one cell, or at one point in time. These 3 sections stay, the red, the yellow, and the blue. But in another cell, the sections that stay instead of the yellow, it might keep the green instead. Those will be 2 different RNA sequences, and when those 2 sequences are translated, they're going to produce different proteins. So these different protein forms are all coming from the same gene that was just edited in different ways, and this is called alternative splicing. And this seems to be very common because humans, we have 20,000 protein coding genes. 20,000. It's not that many. We thought it was going to be a lot more because we produce 500,000 different proteins. So how can we produce 500,000 different proteins from only 20,000 genes? Part of this is for alternative splicing. If a gene can be spliced in multiple ways, then you can produce multiple types of proteins from the same gene. Another thing that happens to the RNA before it gets out of the nucleus is that you need to add a 5' GCAP. So on the 5' end of the mRNA, you add a 5 guanine, modified guanine group, and it's called the 5 GCAP. And on the 3' end, you add a sequence of multiple adenines, like hundreds of adenines are added to the 3' end. And this signals that this RNA is ready to be exported out of the nucleus. So if you don't add this 5' GCAP and this poly A tail, the RNA won't be exported out of the nucleus. So it won't be able to be translated. Now that the RNA is exported out of the nucleus, now we can get ready for translation, and that process, even before it begins, can also be regulated. So one way to regulate the translation of the RNA is by prevent the translation of the RNA. One way to prevent the translation of that RNA once it is in the cytoplasm is by enzymes that remove either the poly A tail or remove the 5' GCAP. So here these enzymes are removing the poly A tail, and this ones here remove the 5' GCAP. And doing that reduces the life of that RNA. So that means that if it's translated, it's only being translated for a very short period of time, so only a few proteins are being made, instead of if the RNA stays in the cytoplasm for a long time, it's going to get translated by multiple ribosomes multiple times so that you will make a lot of protein from it. But if that RNA is broken down quickly, then you won't make as much protein from it. Cells have enzymes that are monitoring any RNA that is present and that doesn't have that poly A tail and that 5' GCAP. It's probably RNA that is coming from another organism, like a virus or a bacterium that invaded the cell, and that RNA is quickly broken down as a defense. But cells use that, removing those tags, and it's a way of regulating gene expression. Another thing that can happen is that RNA that is complementary to the messenger RNA, so that the sequence of short piece of RNA matches a sequence in the messenger RNA. If those two match, they will bind to each other, and that will form a double-stranded RNA. And when you have that double-stranded RNA, again, the cell has enzymes that will find double-stranded RNA and break it down. This is another mechanism the cell has for defense, because when you have double-stranded RNA, most likely comes from a virus. So if you see double-stranded RNA, the cell wants to get rid of it. But cells themselves use this also as a mechanism to regulate gene expression, because sometimes they produce pieces of RNA that are complementary to the messenger RNA that they're also making, and that is called the RNA of interference, and it will bind to the messenger RNA, create this double-stranded RNA that will then trigger the destruction of that RNA. So that is a way for cells to regulate their own gene expression. In other cases, that RNA of interference might come from the external environment. So let's say that the RNA didn't get degraded, it was translated into protein. Now that the protein is made, you still have possibilities for regulating gene expression. So this is called post-translation regulation. This is what happens after the protein was made. And the way cells do this is by using a protein called ubiquitin, which is used kind of like a tag. So proteins that need to be broken down get this ubiquitin added to them, and multiple ubiquitins are added to those proteins, targeting them for a breakdown. So when you have a protein with multiple ubiquitin proteins added to it, they will be recognized by the proteasome. The proteasome recognizes proteins that have ubiquitin tags, and it will break down those proteins into smaller pieces, and it will release the ubiquitin so that it can be reused again. So this kind of works as a cycle. Proteins that need to be broken down will get targeted with ubiquitin. The proteasome will find those proteins with ubiquitin tags, break them down. The ubiquitin will be released so it can be reused to tag other proteins. To summarize, we have multiple mechanisms for regulating gene expression in eukaryotes, while in prokaryotes we had mostly transcription regulation. In eukaryotes, we can regulate transcription just like we had in prokaryotes by preventing RNA polymerase from accessing the promoter or transcribing the coding region. But before that, we can also change the structure of the chromatin and make the DNA pack more tightly around the histones or loosen it up around the histones, and that makes the DNA more accessible so transcription is possible. Once the DNA is loose and it is available for transcription, then we can have transcription regulation by regulating the type of transcription factors that are present on each cell. And since there is separation between transcription and translation outside the nucleus, there is also possibility to regulate the modifications that the RNA is happening. So you have things like alternative splicing, or you can regulate if the RNA is exported to the cytoplasm. Once the RNA is exported into the cytoplasm, you can still regulate what's going to be the lifetime of that RNA, so it can be broken down and degraded either by RNA of interference or by removing the poly A tail and the 5' G cap. And even if the protein is made, you still have the possibility of processing that protein in different ways. So proteins that are made go to the Golgi apparatus, and the Golgi apparatus can add different sugars to them, so they can process them in different ways. And even proteins that are fully active can also be targeted for degradation by using ubiquitin, and they get broken down so they can also regulate the lifetime of those proteins.